Hello, this is Brian Kinghorn and I'm going to tell you about the GMS facility in MateCell. This is all to do with handling non-additive effects, and this means that we don't simply look at the value of two individuals to be mated and take the average. There's something special about those two individuals that gives you a non-additive effect, a deviation from the average. And our classic application for this is Genomic Mate Selection, which is why we called it the GMS facility, but it relates to a range of other compatibility scenarios, and this is just a list of some of those here. First of all, genetic dominance and epistasis. These are classic genetic non-additive effects. And another genetic component is the amount of variation you get in progeny, or the Mendelian segregation variance. So that's where the amount of variation depends on something special about the genotypes of the two individuals being mated, and we can control that. That leads on to look-ahead mating schemes, where we're aiming to make better progress in the future as a function of decisions that we make in the present. We also have animal behaviour compatibility. That's a non-additive effect because some pairs of animals will get along with each other better and lead to a more successful outcome in the breeding operations. Then there's more direct control of selfing in plants, and you can encourage an increase, a decrease, or a targeting of a certain number of cells in your breeding operations, and that may help you to achieve the designs that you're looking for. Also, control of allelic compatibility in plants. If you have that information, you can handle that. And then in general, we have must-have matings and must-not-have matings, and if you can nominate what these are, you can use the GMS facility to help control that. And the last example I've got here relates to transport costs, whether it's zoo animals or otherwise, so that the cost of making a mating between two individuals depends on whether you have to make some transport to bring them together. Now, all these components can be accommodated simultaneously in the same run and with dynamic competition between them, as is the case for a range of other components in MateCell. The key issue here is how we predict progeny merit, and that's normally done by taking the EBVs of the sires and the dams and averaging them to make a prediction of progeny genetic merit. So that information comes from your main data file, so that for every candidate in the data file you have an EBV for each trait. We then go and look at the mating list, work out the progeny merits according to the average of the EBVs, and we present these in histograms for you to manipulate. So this is what's often thought of as general combining ability, but what about specific combining ability, where the specific combinations of males and females has an impact? Normally we use average or additive effects and dominance deviations to predict progeny merit. In general, we're looking at the average of the EBVs plus some form of deviation from that prediction, which is peculiar to the specific sire and dam involved. So how do we implement this in MateCell? Well, we have the main data file with the EBVs, but we also have to have a separate file for each trait that is being handed in a non-additive manner, and these files have the sire candidates and the dam candidates with the deviations included. So this is a file that you construct, one for each non-additively handled trait. These traits are recognizable because they start with the letters GMS, as you can see in the histograms here. So as far as you're concerned, this looks just the same as additively handled traits, and we've got some of them here, and you can manipulate them in exactly the same way. However, the calculation of the values that contribute to these histograms includes the deviations peculiar to each sire and dam in the mating list. So you can see that for many of these non-additively handled traits, the EBVs are 0. An example of that would be when we're looking for Mendelian segregation variance, where we're not looking at the parents, we're only looking at the effect of the genotypes of the parents on variation in the progeny. However, for other traits or constructions, and this is really a construction, where we want to look at the impact of selecting on index minus a weighting on inbreeding coefficient, possibly as a research exercise. In this case, we have the index value as if there were EBVs, and then 100 times inbreeding coefficient would be supplied by you in a file like this. So looking at this a bit more closely, we can see the EBVs for the traits, including the non-additively handled traits, many of these being 0. And for each of those non-additively handled traits, you have to have a separate file which contains the deviations for the sires and dams and the combinations. And how you construct this file is reasonably straightforward. There's a description of that in the manual, and it's very similar to constructing a GRM file, and that's why we call it a canned GMS file, to emphasize that it contains information on candidates only, not the whole pedigree. Now I'll move to a simple demonstration. I've opened the file and started the run. Let's have a look at these new traits. Let's start with the first non-additive trait, which is actually pedigree inbreeding. And you can see that the histogram here is very similar to what we have for progeny inbreeding. The only difference is the fact that we've got a different number of classes for calculating the progeny inbreeding histogram. So if we ask to increase the mean of pedigree inbreeding as a non-additive trait, and we then look at the progeny inbreeding graph, we can see half sibs and full sibs. These are actually the same results coming out of those. This is a very simple demonstration that what we're doing works. We could do something similar with the GRM derived inbreeding coefficients, but let's move on to dominance. So the solution mean for dominance is very similar to the candidate mean. Let's push that upwards. We'll put an increase in the mean with a weighting of 1. And you can see that increases considerably with little change in the frontier. So what we're doing here is largely about mate allocation. Let's increase the push, go up to a weighting of 2, and we can see that there's now a bit of a compromise in terms of the animals selected. There's a decrease in genetic diversity in order to achieve this. And what we can do is push that even harder, and we'll get an even stronger result for dominance, but at a bigger compromise. You can see we've got a very quick evolution of this. We've got multiple different algorithms that compete with each other on the same framework, and one of our integer programming components has done rather well there. You can make the emphasis on this total by taking off the weighting and frontier, put that to 0 and update, and we get an even stronger result. So this is absolutely pushing only for dominance, and this looks like it's the global optimum. So if we go and look at out GMS non-additive and update that, this shows us the actual dominance values that we get for each mating, and that's the thing that we've been pushing hardest. Now we'll move on to must and must not have matings. So let's look at what we have in the GMS files for those two. First of all, must. Everything is 0 except I've got half a dozen cases of a 1 in here, and this is female Dallary. And if I go along to the left, number 8, that's Wayne. So the mating that we must have is between Wayne and Dallary, and there are 6 cases like that. And while I'm here for must not, here we have a mating between Christine and row 35 is Alexander, which must not be had, and we've got a number of those in that file. So let's look and see what happens when we make manipulations based on them. First of all, I'll take the weighting off here and put the weighting back on frontier, otherwise it's going nowhere. And that gets us back to where we were. So with must use matings, I'll simply invoke an increase in that. Let me put a weighting of 10 so we get it. And when we resume, 4, 5, and 6. So if I click print matings and go to the result file, here it is. Update it, and if I go along and look at the must use matings, there'll be 6 matings with a 1 in there, and the last 2 are these matings here between Shepherd, Guyana, and Alistair and Alison. Now if we go to must not, well, we don't have a problem there at the moment because there aren't any must not use. If I did have some, then I would push downwards to get a 0, so that was quite easy to handle. Let's finish by looking at this construct of index minus 100 times f. If I ask to increase that, let me give it a strong weighting and resume. And I'll take off the weighting on the frontier altogether so that this is the only thing that we're pushing for. And you can see that we will have decreased progeny inbreeding. We'll probably see that here. It's come right down, this red line. It's the history of where it's been. And so this is a simple illustration that a very strong emphasis against progeny inbreeding doesn't actually do much to give you increased diversity. We're not far off the top. Now we've got a number of different non-additive traits there, and we can make them compete with each other in the same way as other components in MateCell. And lastly, I'll give a demonstration of a little bit of control over selfing in our simple bisex demo plant example. So here, for those of you who are not familiar with it, this says that individual 11 can be used 30 times as a male plus 30 times as a female. So the non-additive trait I've added there is called GMS underscore self, and all the EBVs are 0, and that's because everything we want to do is to control selfing alone, which is to do with the interaction between an individual and itself. So all the male candidates down here, 10 to 24, are actually the same as the female candidates 10 to 24 because of bisexuality, and selfing is denoted by a 1, and that's of course is just like an identity matrix. So let's go and make the run for that. I'll go and find the file. You'll find this example file in your download package, and here's our one non-additive trait, self, and at the moment there are 4 selfings in there. Let's say we had actually wanted to target 6. Well, we've got 30 matings, so I shall go for tactical desired gain stabilizing, and I'm looking to get 20% of the matings, that's 6, to be self, and we get that immediately. Here we have 6 of these matings are selves. Let's have a look at the results there. And if we go down here and look at those, we would find 6 selfings in there. For instance, there are 2 at the end here, 21. In fact, there's duplicate mating, which if you don't want, you can get rid of as well, because there's waiting for that. Alternatively, if we wanted to have 15 selves, of course, we could just go for 50%. And if I resume, I'll get the result there. And you'll notice in this particular case, we're not getting much compromise on the frontier. And that's because this is largely about managing mate allocations. So I've just given you a smattering of examples. With this control of non-additive effects, you will probably find a range of other applications, and we would be very grateful for any ideas that you can share so we can make that available to others. I'll finish by saying a few words about look-ahead mate selection, which is a term we banded around in the 1990s. More recently, look-ahead mating schemes is also being used. And there's a nice paper by Tobias Nijhoff in GSE. There's a nice illustration that what you should do now depends on how far ahead you want to look. And with this example, with 3 individuals with plus and minus alleles, you can see that generations ahead affect the best pairs to select. Now, I'm not going to illustrate that here. You can pause this and work that out for yourselves. It's quite a nice example. So that paper is here, and Tobias and his colleagues have now generalized this for n generations ahead. And Tobias has kindly contributed a bit to our manual to show how to use the GMS facility to use his formulations. And that's for fixed cross values. That is for where the values you can put into a matrix like this are fixed, and therefore they can be read in at the beginning of an analysis. However, there are some cases where cross values, and probably even just selection values, are dependent on other individuals that are selected. So let's look at that. So for example, if Samir here has some valuable rare alleles, then they're going to be more valuable if they're not represented in other individuals that are selected. And as you tend to select more individuals that contain those other alleles, the value of Samir becomes lower. So this effect is largely about selection, but of course you can consider where you have diploid epistatic complexes that are rare and valuable, and you might want to be targeting these in the future, whether you know it or not. The selections and mate allocations can be important there, and they may be dynamic as they are for selection values. So in those cases, and there are a number of examples from Kath Kemper, Lee Hickey and Saba Moenizad, there will be a need to calculate these values on the fly. That means that every time we make an evaluation within MateCell, we have to work out new values for what's in this matrix. Now that's possible with developments inside MateCell, but what we can also do is interface with other software and calculate these values on the fly using an appropriate portal between components of software, and we've had some discussion with groups on doing this for these purposes. Well, that's us coming to the end of the video. We'd be very grateful for any feedback on other applications that you see for the facility that we've provided you in MateCell, and thank you for watching.